name is Allison Bradbury, and I am a principal investigator in the Center for Gene Therapy at Nationwide Children's Hospital, and I hold dual faculty as an associate professor in the Department of Pediatrics at The Ohio State University College of Medicine. So Krabbe disease, it commonly presents as a really devastating early onset childhood disease with rapid progression, and death often occurs by 2 years of age. But we've known for over 50 years now that the substrate, psychosine, that causes Krabbe disease actually accumulates during the fetal stage, so before the babies are born. We also know from decades of experience with hematopoietic stem cell transplantation and the implementation of gene therapies for other lysosomal storage diseases that early implementation of the therapy is necessary for the greatest benefit. So what we're going to study with this grant is we're going to use the naturally occurring dog model of Krabbe disease to determine when the disease onset occurs during the fetal period, and then deliver a gene replacement therapy in utero to try to really prevent disease prior to the onset in the fetal stage. Rosenau Family Research Foundation was formerly known as the Legacy of Angels Foundation, and they actually provided really instrumental funding during my postdoctoral research days at the University of Pennsylvania when I was characterizing and studying the naturally occurring dog model of Krabbe disease. I was able to utilize the data from my Rosenau Family Research Foundation grants to apply for NIH funding, and was really able to leverage that to launch my independent career. I was able to obtain an NIH F32 individualized postdoctoral fellowship, and then was able to get the NIH Pathway to Independence Award. Therefore, the Rosenau Family Research Foundation funding was really a catalyst that I was able to leverage for larger NIH funding, and has been really instrumental in my career related to Krabbe disease. So I've actually been familiar with Rosenau Family Research Foundation for over a decade now, and we hope to really utilize this current grant to, again, get preliminary data, this time related to the in utero delivery of gene therapy for Krabbe disease, and over the next 3 years, really be able to launch this program and then be able to apply for larger funding mechanisms, again, such as the NIH. So my great uncle, my grandfather's younger brother, was actually a veterinary pathologist at Johns Hopkins, and he discovered GM1 and GM2 gangliosidosis in cats. He then established cat colonies for GM1 and GM2 gangliosidosis and dedicated his career to understanding how these diseases worked and if there were potential ways to treat them. I then, decades later, did my graduate work at Auburn University in the lab of Dr. Doug Martin, who happened to be my great uncle's first ever graduate student. In the lab of Doug Martin, I worked to develop an AAV-mediated gene replacement therapy for GM2 gangliosidosis, also known as Sandhoff disease. We used the cat model to eventually translate this gene therapy to the clinic, and to date, over 10 babies with GM2 gangliosidosis have received this gene therapy. Krabbe disease is very similar to GM2 gangliosidosis. They're both lysosomal storage diseases, and in fact, they're on the same lipid pathway. So when I was finishing my graduate school and I was deciding what I wanted to do next with my career, I wanted to take our learnings from GM2 gangliosidosis and apply them to a really similar and deserving disease. And that's where I landed on Krabbe disease. Krabbe disease also has a large animal model, but it's a dog model. So I went to the University of Pennsylvania to study the dog model of Krabbe disease and develop an AAV-mediated gene replacement therapy. I've been working on that for a decade since, and we really hope to translate a next generation gene therapy to the clinic for Krabbe disease. in the last decade, there have been a lot of advancements in genetic medicine that have allowed the identification of diseases even earlier and more accurately. And with being able to pick up genetic diseases, we now have a real opportunity to intervene with genetic therapies. What gives me real hope is the field of gene therapy and the advancements that have been made in the technology and the implementation. We've now seen the translation of AAV-mediated gene therapy for a number of lysosomal storage diseases and leukodystrophies. We've even had gene therapy clinical trials start for Krabbe disease. The fact that these gene therapy clinical trials are showing an improvement in disease for Krabbe and other similar diseases really gives me hope. I think that we will continue to improve, advance, and optimize these technologies, and they will become even better in years to come. I'm very excited to see how gene therapy continues to improve the lives of kids with Krabbe disease. The best parts of my job are getting to mentor the next generation of scientists, researchers, and physicians. It's really rewarding to be able to spend time in the classroom teaching students about gene therapy. And it's also really rewarding to have graduate students be trained in my lab and see them be able to develop gene therapies that we hope to translate to the clinic. So really being able to teach and mentor the next generation of researchers is one of the best parts of my career. The other best part of my career is actually seeing gene therapies that we've worked on in the lab reach the clinic. It's honestly been the most rewarding part of my career to see a gene therapy that we tested in cell culture and animal models and then did safety studies. And often the road is really long to get to the clinic. But when we dose that first patient and subsequent patients after, it is the most rewarding feeling, and all of the hard work is worth it. The most challenging parts of my job tend to do with having to fund a research lab because we have all this exciting science that we want to do. However, it costs a lot of money. And these experiments often take a long time, especially when we're using animal models and trying to translate something all the way to the clinic. The major funder of biomedical research is the National Institutes of Health, or the NIH for short. The NIH has become increasingly difficult to get funding from, and based on that, it makes running a research lab precarious and uncertain during these times. Another challenging part, specifically in the translation of genetic therapies, is the high cost that comes with manufacturing and performing these clinical trials. Therefore, it's very hard to keep the development of a genetic therapy at an academic institution. So we rely on industry partners such as pharma to come in and help translate therapies to the clinical trials and eventual hopeful FDA market approved drugs. However, with rare diseases, because there's so few patients and we're hoping with a one-time treatment with gene therapy, this is not really attractive for pharmaceutical companies. So it's difficult to get funding to really translate a drug to the clinic and eventual FDA approval. The first advice that I would give for students if they're trying to pursue rare disease research is that it takes persistence. It might be difficult to get the grant funding and to get the papers published, but stick with it. It's worth it. And the second thing I would say is to let the rare disease heroes, the patients and the families themselves, inspire your work. Get to know these kids, what the families go through to live day in and day out with these rare diseases. And that is so motivating. Once you really let these patients and their families drive your research and your motivation, there won't be another field that you'll want to go into. I'm outside of the lab, I have two young sons, so they keep me very busy. Spend a lot of time on the soccer field and the t-ball diamond. However, we also enjoy boating as a family. So we like to do skiing and wakeboarding. We like to travel and experience new places and cultures. And I also play tennis. So I play on a USTA Career League women's tennis team, and that has brought me a lot of joy outside of the lab.